Hi, so I'm the exact opposite of the last speaker. I've been doing cardiac modeling for a number of years and have far too little consideration of uncertainty quantification. So I'm really pleased to be here today and yesterday to actually do a lot of learning. I'm also pleased to talk about the small little simplistic work we've been doing in uncertainty quantification in the parameterization of cardiac action potential models. I'm from Simula Research Lab. We're in Oslo, Norway, and I'm going to talk about the work done with my collaborator Aslak and mostly by the PhD student Carolina Horgmo-Jaeger. So a little bit of a background. We've been working on a project for a little while trying to detect drug effects, and specifically we are working with these little cardiac microphysiological systems. So these are these little beating constructs of heart tissue that are grown on these little microfluidic systems where you can control the environment. And it's a medium throughput kind of device and can get sort of nice measurements optically of the cell dynamics over time. Here they can use these fast dyes and give us good information on the voltage and the calcium in the cells. And now these can be perfused with drugs, and we can see changes to the action potential, changes to the calcium dynamics with increasing drug dose. And our question has really been, can we sort of understand some of these drug effects from the observed changes, specifically because these cells are really still not adult cardiovascular cells. They're still somewhat immature. So we've been working on this for a while, and our approach has been to try to augment this with in silico methods. Can we take these measurements and pass them through a computational layer, invert the measurements into models, and then can this inversion tell us something about what's happening? Can it tell us what channels are blocked and hopefully what we can say about what's going on in adult cells? So the model that we're thinking about are these models of cellular action potential dynamics, and I'll just mention them briefly for the people who might not be familiar. I know a lot of people are really familiar with them, far more familiar than I am, but these are models that depict how the cell behaves when it fires. It gets some stimulus, it's sitting at some polarized state, it depolarizes, and this ends up giving a cascade of reactions that have the cell twitch. So this is kind of the firing of the cell. And this is measured in terms of the transmembrane potential, the difference between the inside and the outside of the cell. And it's built from these currents that go across the cell membrane, a bunch of different species, and we kind of build these models of the dynamics. So we kind of think about the various currents that kind of go in and out of the cell over the action potential, as well as how calcium, which drives the contraction, gets released from intracellular stores and then taken up over the heartbeat. This is a pretty basic ODE system, and we describe our change in membrane potential as the sum of all these inward and outward currents. And all of these individual currents, they can have a lot of different forms. There have been a lot of work going on to parameterize these and understand what's happening with all these currents. They're actually kind of governed by membrane proteins that they can find the structure of, they can test, they can modify. So there's been a lot of work in understanding them. They generally take the form of something like this, where you have some sort of scaling max conductance, you have some sort of open probabilities. It can be a Markov model, it could be some other type of model to describe when they're going to open, and some sort of resting potential that says when they turn on and when they turn off in the cardiac cycle. And these models, there's a lot of these models have been around forever, I guess 50 years or so. And what's often done when you want to try to parameterize them to a given piece of data is to play with these max conductances. And this is not the only way. This is a kind of a naive and simplistic way, but it is the way that's often done. And I was going to put up the quote that Andrew McCulloch had with the number of parameters. But I think I'll pass on that. But there's a lot of degrees of freedom here, and that means that when you change these conductances, you can reproduce a lot of different type of cell behaviors. For example, we can look at one set of max conductances, and we get this standard cardiac action, ventricular myocyte action potential, one spontaneous beat when stimulated, or you can choose another parameter set, and you can get something that more looks like a neonatal or fetal or stem cell derived cardiomyocyte, spontaneously beating, longer action potential. So this is a kind of a common way that people look at these models. They change the parameterization, try to fit these action potential measurements, and then hopefully say something about what's going on. We've been working on this quite a while, and we've been getting lots of data traces from Berkeley, trying to match them, invert them into models, and we can kind of generally reproduce our action potentials and calcium transients, even when we have like very wildly differing action potentials in calcium dynamics. So it's a pretty broad model. Generally optimizing this requires evaluation of a very large parameter space. It's a cheap computation, but you have to do a lot of it. There's many models and methods available to do this. We've been mostly looking now at, can we use this to detect drug effects? So now we want to say, okay, we have some sort of model that represents our cells, and now we apply drug to our cells, and we see some sort of change. Can we find adjustments to the individual currents to minimize the misfit, create some cost function and do some optimization, find the model that fits, and then see what happens. So here's an example. This is some data that we have. It's a series of voltage traces with the application of a drug at escalating dose. And same for calcium. We see kind of this decrease in the voltage in the calcium. Let's build a model. Let's optimize a model that matches this data, and we do that. We can build these models to say what's or to look what's happening, and now we can ask the models what changed. What did we have to change to make the model fit? And this all depends on how you ask the question. Here in this case, we're asking what drug characteristic does it have to have, and specifically what sort of IC50 value does it have to have for all the different channel blocks. And we see, okay, oh, this drug looks like it's a calcium channel blockage with a IC50 of about 38 nanomolar. And we know what this drug is. It's nifedipine, and that seems to work quite well. We can go to literature and say, yes, that's actually what's happening. So we have some kind of ideas that this can work, but we don't want to do this for drugs where we know the effect. We want to do it for drugs where we don't know the effect. And that's when we start having a lot of problems. That's where we've been kind of running into some roadblocks. And a big problem that we've been seeing is the solution uniqueness. We have a lot of big parameter space. We can make a lot of simulations that have sort of similar dynamics. And this is nothing new. People have kind of known this for a long time. Globally, we know that for a given model, multiple parameter sets give the exact same solution. And this was work 4 years ago, where they did a kind of a global search using genetic algorithms to look at across a huge variety of parameter space, and they can make action potentials that are identical, but if you look at the scalings of the parameters, the sort of scaling of the conductances, they're very different. So we know that the models are not unique, and that there are multiple solutions. The other problem, the one that I'll be talking a little bit more about today, is that we really see that there's insensitivity in the local parameter space. So here's an example. We build some cost function and look and see how the model changes as we traverse some of the parameter space. And we see these directions in the cost function where there's very little change. And that's really bad for us because we want to detect all changes. And so if by chance a drug blocked calcium and the hERG channel along this vector, there would be no change to the model, and we wouldn't be able to see anything. So this becomes something that's really not identifiable. And the same thing here. So this is a problem that we've seen, and we wanted to kind of see if we could give some quantification to this, if we could kind of say what's happening. So let's go back to the problem setup. I can say that again. Here's our model, and we have some sort of action potential. Now let's make a measurement. Okay, we see that there's an extension to the action potential, and we do a model inversion, and we've identified that this is a hERG blocker, and that's, I mean, maybe that's a really bad thing, maybe it's not, but we don't know is whether, are there some other combinations there that we don't see? And we also can ask the other question. We have very noisy data. Maybe there's no change. Do we say that this drug had no effect? Or are there combinations of these currents that all can kind of co-occur and cause zero change in the action potential? So we've been really interested in the past year of kind of understanding what can we see and what can we not see. How sure are we? And can we quantify this, some of this uncertainty? And this isn't kind of the real formal uncertainty quantification that I've had been introduced to over the past couple of days, but this is kind of how we can measure what we can see and provide tools that we can help in the development of our own models and methods. So we also, I've been impressed by the complexity of kind of what's been going on. We took an alternative approach and tried to go back a little more simply and really look at our model and the properties of our model as simply as we can. We have some sort of action potential, and we want to see a change in it. This is our equation. What we really want to see is a change in the total current. If we see a change in the total current, we should see a change in our action potential. If we don't see a change in the total current, we won't see a change in the action potential. And the total current is built up from the sum of all these individual currents. Here's the kind of the depiction of how they kind of look over time. So our maybe too simplistic approach, but the approach we've taken is to kind of take a linear look at this system. This looks a lot like a linear combination of systems. We can make a vector kind of describing our total current over specific time intervals. We kind of build this up by stepping through the simulation and finding and binning the current over time. And we can do the same thing with our individual currents in our model. And so we kind of have this linear combination of currents that end up to our total current. And if this total current changes, then we should be able to see changes in our action potential. So can we look for combinations that are going to be visible and not visible? So our approach was then to collect this into a matrix and rewrite it as a matrix vector problem. We have this current matrix. It has our currents aligned in the columns of the matrix, all the individual currents of the model, and they're kind of stepped through in time along the rows. And now we know what our total current is in the same sort of vector form. We have a current matrix. We introduced a parameter called the scaling. This is just the scaling of all the currents from our first current to our nth current, and we can rewrite our total problem as just a matrix vector problem. And what this lets us do is look quite robustly at perturbations of the system, right? I mean, if we have a linear system, we can really kind of understand the perturbations a lot easier and more quickly. If we look at some sort of current perturbation, now some drug will perturb these currents. It might block current 1, it might do nothing to current 2, it might promote current 3. We'll end up with some sort of current perturbation, and now we have a total perturbed current. And what we're really interested in is how large is the effect? What's the difference? What's the size of the difference between our perturbed current and our unperturbed current? And we can kind of just do we can quite quickly see that this is determined by the matrix product of our current matrix with our perturbation vector. And this is just a property of the matrix A. So we start looking at kind of just some tools to evaluate this, and PCA is kind of one. PCA it's very similar. We're just looking at the singular value decomposition, which kind of gives you PCA. Common matrix decomposition is used a lot. One is to kind of understand the directions of this matrix vector product that are going to be large and that are going to be small. Breaks us down into these three matrices, unitary representations of the column and row space, and also this diagonal trees that give the scaling between these two spaces. Just so you can see what we're actually doing, if we kind of had a model that was defined by three currents, we would build our current matrix with our currents in the columns. This gets broken up into these left singular vectors, describing both the column space and the null of A transpose. We have our singular values and our right singular vectors. And the property that we'll exploit is that all of this satisfies the relationship that between these various matrices and columns, where the the matrix vector product of the singular vectors is equal to the singular value or scaling the left singular vector. So what happens is we can consider a perturbation along a singular vector. So now let's take our perturb vector, which is our current scaling plus some scalar multiple of our singular vector. We can write out explicitly what the perturbation will be, and the nice thing is that the size of it is going to be only the scaling factor times the singular value. So this means that we'll expect changes to this total membrane current with perturbations that are associated with large singular values. While if the singular value is very, very small, we'll expect little to no change in our total current, and we shouldn't see any sort of changes in our action potential. So if this was a linear system, this would be true. So our next kind of question is, does this apply to the nonlinear dynamics of these cardiac action potential models? It's a big question. I mean, if these are highly nonlinear problems, and maybe nonlinear analysis would be much more appropriate. We just wanted to see if they would work. So what we did is we did this for 3 common ventricular action potential models. We looked at the Tentusher model, the O'Hara model, and the Grandi model, all kind of used, and we ran the simulations. We built these into these current matrices, did the SVD, found the singular values and the singular vectors, and then proceeded to kind of test these nonlinear systems along these perturbation vectors to see what happened. So if we think about the Tentusher model, it has 12 currents. Our SVD gives 12 singular values with their corresponding singular vectors. And here we can see how they kind of, what they look like. So we have a very high first singular value all the way down. They span 5 orders of magnitude. And here we can see the scalings of the individual currents that these correspond to. So our highest singular value corresponds with the fast sodium current, which is unsurprising because there's nothing that can really kind of compensate for that kind of can make that not visible. Well, then we have all these other sort of singular vectors that are kind of combinations of the different currents. And when we get kind of here, some of them are very intuitive. This is like a background, these are background calcium and sodium currents, so they can balance each other completely and just be invisible. So the question is, does this kind of work in the nonlinear state? So we ran the simulations, and we can sure enough see that the simulations where we vary along the large singular value associated vectors are big. You can't really see this, but this all happens in the upstroke, but that it's still very visible. And then when we get to these vectors associated with small singular values, we don't see a change in the action potential. So if we kind of move through anywhere in this space, we don't expect to see any sort of changes. We can look at this also by using some sort of cost function and see the same thing, that quantify the same way, that the directions in the parameter space associated with large singular values give changes to the action potential, while ones that are associated with small singular values don't. And this gets us to the ability to kind of do some identifiability, and again, this is maybe where we're a little bit naive here, but we wanted to kind of quantify this. We want to say, what does this mean? The nice thing is that the singular vectors provide an orthonormal basis for all perturbations, so you can build any perturbations from this basis. So let's kind of break this into an identifiable subspace, so the subspace of perturbations that we can see changes in interaction potential, and an unidentifiable subspace. When these are all perturbations that we don't expect to see anything in our simulations. And now we can just build an identifiability index that is the orthogonal projection of any perturbation we want to make into these subspaces. So perturbation exists fully in the identifiable space, it would have a value of 1, while if it exists fully in the unidentifiable space, it would have a value of 0. And we can do, for any given model now, we can look at the perturbations along sort of the currents directly, and we can kind of come up with this index of identifiability. So here we can see that a few of the currents should be highly identifiable, well, a lot of them are not very good at all. So, and when we're doing our kind of analysis now, we measure some sort of drug response, we can get sort of at least a gut feel check on whether we expect this to be like a real effect, or there are so many permutations of unidentifiable currents along that vector that it's a bad choice. I'll step through briefly the other two models we looked at. The same thing with the Grandi model. This time it has 15 parameters, so we get our 15 singular values. And again, we kind of can break this all down. We see that we have big changes in the directions in the parameter space that are associated with large singular values, and almost nothing in these perturbations associated with small singular values. So it's this linear analysis, this linear approach to looking at this nonlinear problem gives us at least some insight that we can use. And again, we see this kind of range of identifiable currents and the group of fairly unidentifiable currents. Last one I'll talk about here is the O'Hara model. Again, 13 parameters, 13 singular values. We see kind of generally the same trend. So this is our tool for quantitating the uncertainty when we make predictions for a perturbation. The last thing I kind of want to talk about is using as a tool to understand improvements of identifiability, because this is not surprising. No one who's worked with these models would be surprised at all by this level of quantification of the identifiability of the currents. This is a kind of a well-known problem. It's not surprising, but there's been a lot of effort to improve the identifiability of these models. And so we hope this also can be a tool for understanding how to develop improvements of it. So let's look at our Tentuscher model. We can see that our second singular value has big changes associated with but our last one has 0. And unfortunately, using this single action potential waveform, we have almost no change even in this 6 singular value. So our unidentifiable subspace is very large in comparison to our parameter space. And this means that we have to have a lot of unidentifiability in the model. And we see that in our quantification of that. But some very smart people have seen things before, and for example, in the same paper I showed earlier, they noticed that when you sort of change the pacing timing to kind of maybe look at some of the inherent frequency dependencies in these channels, that they could and they tried to match this waveform, they could really reduce the error associated in their parameterization, and all their kind of parameter choices narrow down in space. So our question is, will linear analysis show the same sort of trend? So let's look at that. Here we have kind of a alternative pacing train. We have our pacing at various times. We'll build this whole thing into a current matrix and do the exact same SVD analysis. Again, we see that we have like early singular values that are high and have high change in the waveforms, and our last one still you see basically nothing. But now we can see that even in the 10th singular value of a 12 parameter set, we're having a significant change to our action potential model. And that means we can get identifiability much higher. So this is it shows that we can kind of look at these techniques and try and see what's identifiable and what's not. Another nice thing is we can do this kind of on a model-specific manner. So these are all kind of uncertainties associated with a given model. We can look at the Grandi model, do the same thing. Here's what the identifiability is with a single beat analysis. If we do this kind of random pacing, we improve it in almost all channels, but we have some that are somewhat missing. If we look at the O'Hara model, we do the same thing, and here we can see that we get really good identifiability of some, but still some are completely invisible. So this becomes a tool to look at your model, to look at your protocol, to try and see what you should be able to see, what you shouldn't be able to see, and hopefully some kind of level of assurance of the conclusions that you make. So that's it. I think I'm okay on time. Surprisingly, we found that linear analysis gives us some good insight, we hope, into their nonlinear behavior. This SVD approach gives a fast, it's very local, that's one of the drawbacks, evaluation of the visibility of current combinations, and it's also fast. You run these models, all you have to do is collect the currents as you run them into a matrix and run an SVD. It takes seconds per current that you want to kind of do. So there's very little overhead to get this kind of insight. And we hope this method is useful across a range of models and protocols to provide some sort of quantification of uncertainty. With that, I'd like to acknowledge Ashok and Carolina, as well as our collaborators at UC Berkeley, and I'm happy to take any questions.
Hi. Have you thought about how much it might improve identifiability if you looked at paired voltage and calcium?
Yeah, we have that. We've actually been doing that. That's the next round, is to have them at the same time. And we thought it was going to help a lot more than it does. That there's still sort of these variability that we kind of can't account for. But one of the big problems with that is that the measurements of calcium are relative. So we don't actually have an absolute value for calcium, and that introduces some kind of scaling problems. So we're working to get these absolute measurements, which we think when we have those, then that we think will give a much better sort of single beat sort of identifiability. Yeah.
So presumably when you try and back out the IC50s from the different models, you get some interestingly different answers.
Interestingly different answers, yeah. Yeah, the models have very different sort of behavior, right? And we should be aware of that there's, yeah.
Any thoughts on what to do about that?
Well, we're trying to make our own model that we have kind of confidence in, that we know what the identifiability, where we can make judgments and where we can't. So that's kind of our approach. But there has to be some nice data sets that everyone could use, that we could kind of do evaluation of all these models. I know the type of data that you guys are building will be invaluable for that. So I think it'll just take some time to kind of shake out what's the right model for the right system, right? And here we're only looking at these max conductances also, and I mean there's the whole other can of worms of the kinetics and where we need to go with that. So there's a lot of big problems still. Yeah.
I have a question. Maybe it's specific, or I just missed that. How is the random pacing done? So how can I imagine that during one simulation it's just random pacing cycle length, or?
Yeah, exactly. Yeah. So it's just I can't remember the exact way it was done, but there's some sort of random interval that was added to. So it was between some length of 0.7 and 1.2 seconds or something. So it was yeah.
Yeah, I think it's like very interesting that this linear theory works here. But I wonder, there are also other types of linear theories, for example, for limit cycles. And did you thought that if you use, for example, periodic pacing, not random, but periodic pacing, and just plot these Poincare maps and use something like what is called, I think, for K theory, so it will give you additional information.
Yeah.
About stability and some.
Yeah, exactly. I mean, the instability of these solutions is another big kind of thing that we've been looking at, and yeah. So I think there's a lot of mileage there too. You're right. Yeah, definitely.
Thank you very much. Very clear presentation. I would like to ask two questions, if I may. Firstly, I've kind of noticed that you listed IQ1 as unidentifiable parameter, which actually surprises me, because firstly, what can compensate for IQ1? Secondly, using genetic algorithm-based approach, we could identify IQ1 quite precisely, up to several %, I would say. And second question, I'm extremely interested if you tried your single value decomposition approach to something more complicated than nifedipine, something like beta blockers, for example, which would affect bunch of currents, and not only conductivities, but would shift activation curves as well.
Yeah.
So can we identify those?
I don't think we can at the moment, to be honest. For your first question, this is very model specific. I mean, the identifiability of these currents change depending on where you're in the parameter space. So for example, we're just using the published values of the parameter space for these models. And that is the local identifiability. If you have your own model that you fit to data, and somehow you've upscaled the K1 current or something, it could be very different. So I can't exactly say why you can identify K1 in the model you're using, but I bet if we look closely together at the two models and what they were showing, the nice thing is you can just look at those vectors and you can see where K1 is being compensated for. So I would be interested to see what's happening there.
I'll probably ask you after.
Okay, yeah.
Thank you.
Okay.
Okay.
So, well, we've got coffee now anyway, so anybody who wants to follow up on any questions can do so. But can we thank the speakers again?